Welcome to Cell Collective, a collaborative modeling and simulation platform for life sciences researchers, students, and educators. This brief video will demonstrate the innovative tools and features available on the software. After logging in, the home screen displays a collection of all the available models. The models are cataloged into published, personal, and shared models. Published models are fully annotated and peer-reviewed models made available to the Cell Collective community for simulations and further contributions. Models may be browsed or searched by name, description, or tags. Opening a model takes users to the overview page, which contains a brief description, author information, and references. Each component of the model is listed in a tabular format in the Components table. The overview page also contains a network visualization graph, which is an ensemble of all model components and their interactions, displayed within a single network view. Selecting any component brings up its knowledge base, a dedicated page containing a description and detailed annotation of interacting partners, with supporting evidence generated from laboratory research. A key innovation of Cell Collective is the ability to transform this static knowledge and data into a dynamical model, enabling scientists to visualize and simulate biological systems as a whole. The simulation feature in Cell Collective is designed with simplicity and intuitiveness in mind. Biological processes, represented by components that make up the internal machinery of the cell, are simulated in external environments which drive the dynamics of the system. External components correspond to various extracellular signals such as growth hormones and various ligands, and thereby represent the external environment. Once the simulation is started, the activity network displays the global activity level of the virtual cell. Individual components, such as the survival protein AKT and the regulator of cell growth ERK, may also be selected for visualization on the simulation graph. An innovative feature of Cell Collective is the ability to visualize the dynamics of any model interactively and in real time. Using the simple sliders, the amount of each extracellular signal may be changed and the effects on the dynamics of the virtual cell visualized while the simulation is running. As you can see, increasing the growth factor EGF causes higher activation of AKT. Similarly, users may introduce biological mutations to simulate loss of function and gain of function experiments while watching the dynamics of the cell change as a result of the mutation. For example, the effects of constitutively active RAS on the dynamics of the cell may be seen by introducing a gain of function mutation to the RAS proto-oncogene. Elevated ERK activity as a result of a RAS mutation, as shown by the simulation, has also been observed in real cells, further adding to the validity of the model. For user convenience, simulations may be paused, resumed, or ended at any time by using the simulation controls. Models in Cell Collective may also be simulated and their dynamics visualized and analyzed in hundreds or thousands of extracellular environments using the Dynamic Analysis tool. This powerful feature allows users to identify functional relationships between extracellular stimuli and various components of the virtual cell. Similar to laboratory experiments involving real cells, the model requires the setup of extracellular conditions using the dual activity sliders. Once the in silico experiment is completed, dose-response curves may be generated to investigate the dynamics between any components. A significant advantage of computational experiments using this tool is that users may generate any number of analyses without rerunning the entire experiment. More importantly, the dynamic analysis feature allows researchers to generate new hypotheses that may then be tested directly in the laboratory.